Welcome. We discuss IMViC tests. IMViC tests are a group of individual tests used in microbiology lab testing to identify an organism in the coliform group. A coliform is a gram-negative aerobic or facultative anaerobic rod that produce acid and gas from lactose within 48 hours. The presence of some coliforms indicate fecal contamination. The term IMViC is an acronym for each of these tests. I is for indole test, M is for methyl red test, V is for bogus Proskauer test, and C is for citrate utilization test. The lowercase I is for the pronunciation. Indole test. This is a biochemical test performed on bacterial species to determine the ability of the organism to convert tryptophan into indole. This is performed by a number of different intracellular enzymes, a system generally referred to as tryptophanase. When indole is combined with Kovacs reagent, the solution turns from yellow to cherry red. In this, the Kovacs reagent is used that contains hydrochloric acid and para-dimethyl aminobenzoylate dissolved in amyl alcohol. Amyl alcohol is not water soluble, so the red coloration will form in an oily layer at the top of the broth. Requirements: tubes with 5 ml sterilized peptone water, bacterial suspension provided which is of 18 to 24 hours old, Kovacs reagent. Procedure: take a sterilized test tubes containing 5 ml of peptone water, inoculate the tube aseptically with the provided bacterial suspension. One control tube without provided bacterial suspension is also kept. Incubate the tubes at 37 degrees Celsius for 24 to 28 hours. After incubation, add 0.5 ml of Kovacs reagent to the broth tubes. Then observe for the presence or absence of the cherry red ring at the top of the water. Recording of results. The indole positive, it is occurring in case of the Escherichia coli, which shows the cherry red ring at the top of this peptone water, and indole negative, it is observed in case of the Enterobacter aerogenes. Here there is no cherry red ring at the top of the peptone water formation. Next test, that is a methyl red test. This is commonly known as MR test and is used to determine the ability of an organism to produce and maintain stable acid and products from glucose fermentation. The MR test and VP test are performed simultaneously because they are physiologically related and are performed on glucose phosphate broth. Here, glucose fermentation gives the mixed acids and this lowers the pH of the broth. The MR indicator is added after incubation, which is red at the pH 4.4 and yellow at pH 6.2. If the organism produces large amount of organic acids as formic acid, acetic acid, lactic acid, succinic acid from the glucose fermentation, the broth medium will remain red after the addition of methyl red, a pH indicator. The MR negative organisms further metabolize the initial fermentation products by decarboxylation to produce neutral acetyl methyl carbinol. This is also called as acetoin, and this results in decreased acidity in the medium and raises the pH towards neutrality, that is the pH 6 or above 6. Those organisms which do not produce the acid and products, the broth medium will change to yellow coloration indicating negative test. Requirements: tubes with 5 ml sterilized glucose phosphate broth. Next, number 2, provided bacterial suspension of 18 to 24 hours old, and number 3, methyl red reagent. Procedure: inoculate 5 ml glucose phosphate broth with a pure culture of the organism provided. A control is also to be kept. Incubate at 35 to 37 degrees Celsius for a minimum of 48 hours. After incubation, add 5 to 6 drops of methyl red reagent. Then observe for the color change in the broth medium. Results: The MR positive, these are showing red color after incubation, and the MR positive observed in case of Escherichia coli. MR negative, these are showing yellow coloration of the broth and occurs in case of the Enterobacter erogens. Voges-Prosker test. Voges and Prosker in 1898 first observed the production of red color after the addition of potassium hydroxide to the cultures grown on glucose phosphate broth. Harden later revealed that the development of the red color was the result of acetyl-methyl carbinol production. In 1936, Barrett made the test more sensitive by adding alpha-naphthol to the medium before adding potassium hydroxide. Requirements: tubes with 5 ml sterilized glucose phosphate broth. Number 2, provided bacterial suspension. Number 3, 6% alpha-naphthol solution, and number 4, 40% KOH solution. Procedure: take a tube containing 5 ml sterilized glucose phosphate broth, inoculate in it suspension of the provided bacteria. Keep a control tube without inoculation of the bacterial suspension. Incubate the tubes at 37 degrees Celsius for 48 hours. After incubation, add 1.2 ml of alpha-naphthol solution and 0.4 ml of 40% KOH solution. Shake the tubes and observe the development of the color. VP positive is observed in case of the Enterobacter erogens, which is showing the red coloration. VP negative Escherichia coli is observed, and this is the yellow colored after incubation. Citrate utilization test. Citrate utilization test is used to determine the ability of an organism to utilize sodium citrate as its only carbon source and inorganic ammonium salts as its only nitrogen source. When the bacteria metabolize citrate, the ammonium salts are broken and ammonia it is released, and this increases alkalinity, turning the bromthymol blue indicator from green to blue. This is the reaction. Citrate, it is converted into the that is the ammonium salt broken down to ammonia, and this increases the pH, that is towards alkalinity. Therefore, the bromthymol blue, which is initially green, it changes its color to blue. Requirements: sterilize the semen citrate agar slants, provided bacterial suspension of 18 to 24 hours old. Procedure: inoculate semen citrate agar lightly with the provided active culture of bacteria. Keep a control semen citrate agar slant without inoculation of provided bacterial suspension. Incubate at 35 to 37 degrees Celsius for 48 hours. Observe the growth and the development of blue color, denoting alkalization. Recording of the result. The positive, it is in this, the growth on the medium with or without change in the color of the indicator, it is there. Growth typically results in the bromthymol blue indicator turning from green to blue, and the test is negative if there is no growth. Thank you.